Dear students, in today's class we will discuss about the topic transcription in your chapter Molecular Basis of Inheritance. In the chapter Molecular Basis of Inheritance, we concentrate on the basic role of DNA as the genetic material. We are all knowing that DNA can code for the characteristic features and can transfer from the parents to the offsprings in the form of the characteristic features which is expressed in the form of protein. So when we look at this kind of transfer of characters, we have to go for certain basic principles. And the most important basic principle on which this transfer of character by the DNA is taking place is illustrated by Francis Crick's central dogma in molecular biology. When we see in 1958, after the discovery of the molecular structure of DNA by Francis Crick and Watson, Francis Crick alone postulated the central dogma in molecular biology in 1958, which can completely show how the genetic characters are transferred from one generation to the next generation, or how the basic coded messages in DNA is converted in the form of a functional product, the so-called protein we say. So when we look at this central dogma, we can see three distinct phases. In the first phase, you can see that the DNA can increase its number. We call it as DNA replication, which takes place at the time of cell division, replication of DNA. And in this you can see DNA is making its own copies. That is why you can see an arrow mark is representing again towards the DNA. So this can give you the idea that the double helical DNA is making its own daughter DNAs. That is the first phase of the central dogma, the replication of DNA. Now this DNA is carrying many messages for the physical characters as well as the genetic characters. These messages are coded in DNA. That is why you call DNA as the blueprint of the life. So this DNA now has to reflect and produce the functional product, the protein. For this protein synthesis, mainly two phases are there. The first phase is called transcription and the second phase is called translation. The transcription is the process by which the DNA is transferring its messages into messenger RNA. The transcription, which can show the flow of genetic information from DNA to messenger RNA, which is fully represented in this diagram. You can see a complete cell which has got the nucleus. This nucleus is carrying the genetic material DNA, which is coded with many messages for the synthesis of protein. So this DNA molecule now is splitting into two strands. One strand is termed as template strand. The other strand is termed as coding strand. So we have to give much emphasis to this point. The whole thing is happening inside the nucleoplasm. So you can say that transcription takes place inside the nucleoplasm, in which the DNA is making its messenger RNA. And for this process, first of all, the two strands are getting separated. one will act as the template, the other will act as the coding strand. Now, when you see the word coding strand, you can easily understand that something is imprinted in that. Some messages are coded in that. What kind of message is coded? The message for the synthesis of a particular protein. We are all knowing that this coding strand is the real strand on which the messages are coded. Now there is a question, how these messages, in which form these messages are coded? You have studied that the DNA strands are, both polynucleotide strands are made up of nucleotides. Nucleotides basic components are nitrogen bases. Suppose this is adenine, thymine, cytosine. Here also you are getting the complementary pairing thymine, adenine, guanine. So this way the complementary base pairing happens. So when these three nitrogen bases, for example adenine, thymine and cytosine comes together, you are getting a triplet code. A triplet code. Here this way three nitrogen bases will come together and make a code. You call it as triplet code for making each amino acid. For example, this will represent amino acid 1. This will represent amino acid 2. Like that, many amino acids, when comes together with the polypeptide chain, we are making the functional protein. So when you look at this coding strand, which is carrying the exact coded message, you are making the functional protein. But when you look at here, you can understand that the template strand is the one we are taking into our consideration for making the messenger RNA. There is a contradiction for that. I have drawn this particular piece into an enlarged form, and this is what we call the transcription unit. The whole structure put together we can call as transcription unit. Transcription unit is the segment of DNA which undergoes transcription. And when you look at this transcription unit, you can see three distinct regions. The first region is promoter gene. This promoter gene is always at the 5' end of the coding strand. You always refer the polarity in reference with the coding strand. This is coding strand which runs in 5' to 3'. So at the 5' end of the coding strand we can see promoter gene. Promoter genes are always the sites at which a very important enzyme is fixed or will be complex. The name of the enzyme is RNA polymerase. Why we call it as RNA polymerase? Because we are making the messenger RNA. One by one the nucleotide should come in a polymerized chain. So the enzyme which can catalyze this polymerization process is RNA polymerase. This RNA polymerase will come and attach at this promoter site. Promoter site will be followed by the real functional part of the transcription unit, that is the structural gene. Structural genes start from this particular point and ends in this particular point. Structural gene, as the term refers, this is the particular piece which will really transcribe into the messenger RNA. So from here the messenger RNA start, here the messenger RNA ends. That means the real messenger RNA is formed only from this particular limited piece. So you call this particular region as the structural gene which can transcribe into messenger RNA. And now we have the third region, you call it as the terminator gene, the terminal gene or terminator gene. So this is the region where the transcription process will end. This hold the end signal. So the promoter gene hold the start signal by joining with the RNA polymerase enzyme, and the terminator gene will hold the stop signal where the transcription process will be stopped. But in between the real and the functional structural gene which will make the messenger RNA. Now when you look at these nitrogen bases, you can see that the template is giving all sorts of the regulatory rules for the messenger RNA formation. Even though the name of the strand is coding, and this coding strand is the real one which should prepare the protein, here in a contradictory version, the template is directing. Let us see what is the reason behind it. When we see the replication, you have seen that both the parental strands are acting like the template and both will be making the daughter DNAs. But here you will see another thing is that only one strand will be making the messenger RNA. The other strand will not. So there are two reasons behind why only one strand of the DNA will direct the messenger RNA synthesis. Reason number one, when the template is synthesizing messenger RNA, the real functional protein is made, because when you look at these nitrogen bases, you can see that ATG, adenine, thymine and guanosine. The complementary pairing is thymine, adenine and cytosine, which make one amino acid. The second, guanosine, thymine and adenine. Their corresponding is G for C, T for A and A for T. So complementarity is established between the two DNA strand. But here what happens is that if both are making messenger RNA, what will happen? This particular coding strand will go with another messenger RNA and this will be going with the second messenger RNA. So what will happen? Two sets of amino acid chains will form. That can give a confusion to the final protein synthesis. We need only specific and one particular type of protein. And as a result, you can see that since there is a similarity between this coded messages and the messenger RNA codes, you can very clearly see that always RNAs will hold uracil instead of thymine. That part is very critical. So you should always remember that thymine is replaced by uracil. So when we synthesize messenger RNA, always uracil will be taking in place of thymine. So you can see what is the similarity between this particular messenger RNA and the coding strand. You can see thymine will be replaced by uracil. Adenine will be same as that of adenine. Cytosine will be same as that of cytosine. Second one. G, guanosine will be same as that of guanosine. Thymine will be replaced by uracil. Adenine will be same as that of adenine. So when you compare this messenger RNA and the coding strand, you can understand that the messages coded in this coding strand in the form of this triplet code is exactly same as that of the messenger RNA. That means you are preparing the functional protein as the coded messages of the coding strand. But coding can't make the messenger RNA at this point. For that, template will be making its complementary strand as messenger RNA. I hope children can understand what is the basic principle behind it. Once again, coding can never make a messenger RNA. Coding strand can never make a messenger RNA. Why? If coding is making a messenger RNA, the amino acid the sequence will be different. But if template will be making the messenger RNA, the same coding sequence will be found repeated in the messenger RNA, and you are making the functional protein. So from this, you are understanding what is the first reason behind why only one strand is transcribed. The second reason is that when two messenger RNAs suppose form, these two messenger RNAs will come close to each other and makes the coil and can land up in the formation of double-stranded messenger RNA, which will not be translated into proteins. So we have seen all as the transcription. So from this you should understand the three parts. The promoter site or the promoter gene for the attachment of or the complex formation of RNA polymerase enzyme. Second is the structural gene, the definite part which make the messenger RNA, and the terminator gene which stop the formation of the transcription, the messenger RNA. And you can also see one thing, that always we are making the parallel messenger RNA in relation with template, because RNA also can synthesize in 5' to 3' direction. So RNA's polarity is here 5' and here 3'. So RNA polymerase will also progress towards 5' to 3' direction. So I hope that why the two reasons behind why only one strand is getting transcribed. Now, when the transcription takes place, you can see that this particular RNA formed is a nascent RNA. That means immature RNA. We have to make this immature RNA into a mature RNA. For that, we need to do certain procedures. When we look at eukaryotic DNA as well as prokaryotic DNA, you can see that the DNA or the segment of DNA you call as genes. So you have studied that genes are the definite segments of DNA, and in a modified version, genes are technically called cistrons. The genes are scientifically or in a chemical way we call it as cistron. Cistrons are of two types, polycistronic and monocistronic. In the case of eukaryotic organisms, the cistrons are always monocistronic. What is the meaning of monocistronic? Mono stands for one. That means one structural gene, right, will produce one protein. One structural gene will produce single protein. But in the case of prokaryotic organisms like bacteria, the cistron is polycistronic. That means you can see many proteins are formed from a single stretch of messenger RNA. A single messenger RNA can make many proteins. That is why it is called polycistronic. Now when you take this DNA making this messenger RNA, you can see that the formation of this messenger RNA, the immature messenger RNA, has got many unwanted segments. So don't think that we are getting the final refined product. No. In the first stage, it is not having only the useful part. So all those parts which are non-coding, some pieces are non-coding. They are called intervening sequences or introns. I have given I1, I2, I3, I4. That means 4 useless pieces are there. 4 non-coding sequences are there, right? In between these introns, you can see E1 exon 1, E2 exon 2, E3 exon 3 and E4 exon 4. What are these exons? They are the real coding sequences. They are the real coding sequences for the amino acid. When these amino acids will be forming, we can make the real protein. So, when we look at the immature RNA, messenger RNA, 2 distinct regions are there. Exons, which are the coding sequences, and introns are the non-coding sequences. Now we require only the exons. Introns we have to cut and throw it away. So for this cutting, there is again a procedure. You call this procedure as RNA splicing. You know the word splicing. Splicing means slicing or cutting. So when you are cutting, you can see that small cuts are formed, and because of this cut, you can see all the introns are making the loops, small curved loops. These loops will be finally pinched out. That means it will be separated out, and the final matured messenger RNA is formed at the end of this splicing only with exons. Here exon 1, here exon 2, here exon 3 and here exon 4. No more introns. So final messenger RNA now we have made is this mature messenger RNA. Once the messenger RNA is formed, we have prepared that matured messenger RNA. This matured messenger RNA will be leaving the nucleoplasm and come to the cytoplasm. It will quickly go to the machinery of protein synthesis we call as the ribosomes. Many ribosomes at that particular point will come together and make a chain. When many comes and line up, you call it as polyribosome. For lining, there is a thread, the newly formed thread. What is that newly formed thread? Messenger RNA. So messenger RNA holding all the sequences, 3 triplet codes. The first triplet code for amino acid 1, the second triplet code for amino acid 2, the third triplet code for amino acid 3 are all lined up at the polyribosome, and slowly the next and the last phase of the central dogma is happening. That is the translation. So the messages are now translated for the production of these amino acid chain. So amino acids are all scattered in the cytoplasm. These are the scattered amino acid. These scattered amino acid will be brought by another set of RNA. You call them as transfer RNA. So the transfer RNA are mainly responsible for bringing the correct coded amino acid as per the codes on the messenger RNA. So messenger RNAs are carrying 3 triplet codes, 3 nitrogen bases forming the triplet codes. So these triplet codes will be taken by the transfer RNA for bringing their corresponding amino acids. So amino acid 1, amino acid 2, amino acid 3, like that a chain of amino acids are forming. That process is what we call translation. And finally, what protein you should prepare in your cell, is under the control of DNA. So from this we can understand that the central dogma is showing the flow of genetic information from DNA to the final product in the synthesis of protein. I hope that now the three processes are taken into your consideration for the first one, the replication, second one, the transcription, and the third one, the translation. Children, these three major events which is illustrated by central dogma is the major events by which the whole genotypic as well as the phenotypic characters are expressed in our body under the control of DNA. That is why we call this DNA as the genetic material. I hope that the points are all clear, and thank you.